to another lab for advanced exercise assessment and prescription. So today we'll be looking at lab 4, which is regarding the determination of anaerobic power and capacity. So for the purpose for our today's lab, there's multiple purposes, but broadly we're going to be looking at discerning the best ways to measure selected parameters related to athletic performance. So this will be more specific. It's learning about administering field tests appropriately when we want to replace laboratory techniques. So if we're not able to do that lab technique and we want to do this in a field setting. And thirdly, understanding the risks associated with anaerobic testing and what populations or individuals we may or may not do anaerobic testing with. Fourthly, to understand the different energy systems that are involved in anaerobic metabolism, and understanding how to use assessment data when prescribing training or exercise. So just some background theory before we get into the tests. There's many sports that require repetitive all-out bursts of physical activity, and these can range from very short for up to 2 minutes. So things like shot put is one all-out burst of activity, whereas something like also like the 100-meter sprint is again going to be basically an all-out burst, but again a little bit longer. And then something like the Super G is going to be around 90 seconds, but then we still consider this to be a primarily anaerobic sport. So there's no gold standard assessment of anaerobic capacity and power in the same way that for aerobic power or capacity we have VO2 max tests with the direct testing. So there's nothing quite like that for anaerobic testing. So then if we look at where this metabolic side of things, so we know that our resynthesis of ATP in the muscles can occur in two ways anaerobically, so that ATP is going to be used to produce the movement that we require. So one way will be through what's known as alactic anaerobic metabolism. So alactic basically not requiring any, or not producing any lactate. So this combines ADP that's been used with creatine phosphate in order to form ATP once again. So this system, this alactic system, will only last around 7 to 10 seconds before being depleted, and it doesn't require any oxygen. So this system would be what we can kind of see up here as the light green in this graph. So this graph is of our ATP turnover rate. So there's the amount of ATP that's being produced through any multiple energy systems. So that light green is fairly depleted within sort of like in 6 seconds in this graph, but somewhere in sort of like 7 to 10 second mark, we're using up most of that phosphocreatine source. Which our next source of anaerobic metabolism is through lactic anaerobic metabolism. So that's going to be producing lactate. This involves the breakdown of glycogen through glycogenolysis or blood glucose through glycolysis, which results in the formation of lactate. And so this will occur in the presence or within the absence of oxygen and can last for up to 60 to 120 seconds. So this figure here, that's sort of this medium green. This figure only goes up to 30 seconds, but we can still picture that this is still going to be producing ATP continuing. And then just to note, on the bottom here is dark green, so this is going to be our aerobic metabolism. And so this will be, even at the end of 30 seconds, we're almost up to like 50% of our ATP production is being produced through aerobic means. So now we want to get a little more specific into what the capacity and power side of things mean. So we'll just start sort of a breakdown through alactic anaerobic capacity. So again, this is our alactic phosphocreatine without lactate being produced system. And if we're talking about capacities, this is going to be the total energy output that the system is able to produce. So over that like 10 seconds or whatever it happens to be, the total energy output that can be produced. Then if we move down to the power for that same system, this is the maximum rate of energy output that the system can use. So we can kind of think of the power as the highest amount of power we can produce, or sorry, the power, as we can produce, as that system maybe can produce, and then the capacity is the total amount of energy that can be produced. Then we have the same sort of thing for our lactic anaerobic system. The capacity will be for an effort lasting 60 to 120 seconds, the total energy output that system can produce. And then the lactic anaerobic power will be the highest power output that can be achieved. That's not including the alactic system. So generally after the first 15 seconds when that alactic system is depleted, we want to look at the lactic anaerobic power. So why do we do field testing? There's no need for any sort of expensive laboratory equipment. We can test a number of participants at a time, so we can test an entire team at a time if we want to do that, and then it's generally inexpensive to administer. The validity is questionable for some tests. Tests need to be valid in order so we know that we're actually measuring what we want it to measure. So field tests not generally as accurate as sort of like the criterion or the gold standard test that they're designed to replicate. So this could be something not necessarily in the anaerobic side of things, but like doing the beep test to assess your aerobic fitness in comparison to doing a direct VO2 test. It wouldn't be as accurate as sort of that direct test, but it's a lot less expensive to do and simpler to do. There are many field tests that have been validated, and there's formulas that have been developed in order to accurately determine work and power output that are similar to laboratory measures. So the 5 tests that we'll be going through today are the cycle ergometer test, or the Quebec test, the 20-yard shuttle, a vertical jump test, Margaria-Kalamen test, and a standing long jump. So we'll start off with the Simoneau cycle ergometer test, or the Quebec test. But before we get into that, we'll just look at our participants, their general information. So all their information is listed here. So the height is 1.77 meters, 67 kilogram body weight. Date of birth and sex are listed here as well. And so as you can tell, I'm the participant for this lab. So we'll start off with this cycle test, and we'll look at the participant. They're getting their legs going as fast as possible with no weight on, and then when they're pressing that button, that's dropping resistance, and they're just going to keep cycling as fast as possible. So if we walk through kind of the steps for this while they're going as hard as they possibly can, first step is going to be making sure that the athlete is fully warmed up on the bike with at least 2 short sprints of like 5 seconds max, just to make sure that they're able to get their legs going as fast as possible. Our next step, following that warm-up, remove all the resistance, and then the athlete will cycle as fast as possible with no resistance, just to get their legs moving. And then once they've reached their highest cadence, resistance will be added, and the athlete will cycle as fast as possible for 90 seconds. So when we press that button on the cycle ergometer here, that's when the resistance was added. And so that resistance for that test will be at a rate of 0.03 kps per kilogram of body weight. So just to give you a reference, comparing this to the Wingate test you guys would have done, that was 0.075 kps per kilogram of body weight, so a little over twice as much weight. So this one's a lot less resistance, but it's going to be for longer. So this test, similar to the Wingate, you would have completed an exercise physiology, but 90 seconds instead of 30 seconds for the Wingate. So just trying to keep the legs going as fast as possible for 90 seconds. And then if it's completed, so the computer should automatically stop if we've got it set up right. And then we want to make sure that we're giving our participants a few minutes of just easy cycling to cool down. Generally for this one, you don't feel quite as nauseous as you might for the Wingate, as it's a lower resistance and there's going to be a lot more of an aerobic contribution rather than just that all-out high peak power type sprint. So we want to let our participant cool down, and then we can look at the results. Alright, so these are some of the results that might be shown. So for this individual, we're looking at watts per kilogram in this case. We also do have our peak power in watts there, and average power, and our minimum power. So there's no published normative data for the Quebec test to specifically compare to. So you kind of can compare still within an athlete over time, or you might be able to compare with some of like your own historical data. So our next test will be the 20-yard shuttle, and I'm just going to pop off screen for a second so that we can show the setup of it. So we have the athlete starting in a 3-point stance. They will then sprint to one side, and then they'll turn, touching the line with their foot, sprint back to the other side, turn once again, touching the line with their foot, and sprint through the center line. And then the best of the two times will be recorded. So we'll just play that. So you can see them starting in a 3-point stance, start the timer, turn, foot touches the line, and then back through and sprinting through the line. So nice and simple test, really easy to set up. And then if we're looking at the results of this client's two trials, we can see they're fairly similar, and then you're going to take the best of the two trials for when comparing to normative data. So in this case, we don't have necessarily general population normative data, so we can compare to this trial that gives you a percentile rank, and then also for different sports. So you can decide one of the men's ones. This athlete clearly is below for all of these, so you can discuss that. Alright, so go to the next. Alright, so the next test is going to be a vertical jump test, or the Sargeant vertical jump. So the height of the vertec, which is going to be the stand that we have here, is the sort of commercial name for it. The vertec color veins can be adjusted so that the athlete's fingertips touch the bottom vein while standing flat-footed. This makes it easy for doing any sort of math after of calculating the vertical jump. Then, if the athlete you expect is going to be able to actually jump higher than the colored planes, you can set the colored planes to be above the athlete's fingertips. So maybe you'll set them to be 30 centimeters above or a foot above the athlete's fingertips, and then you just have to take this into account when you're calculating the vertical jump height. So then the athlete will perform a counter movement, so swinging their arms back, and then from there they'll jump and tap as high as possible with the dominant hand, and then we'll take the best of 3 trials. So they'll do this, have a few minutes to recover, because we know that phosphocreatine system needs time to recover in order to fully be able to jump as high as possible again, and then we'll record their best trial. So how we actually record the trial will be by using the distance between the neighboring veins. So these little markers on the vertex system. So between each vein, between the blue and the white veins, so it's hard to see the white ones, but there's white ones here. So between those is 0.5 inches, and between like blue to blue or white to white is going to be 1 inch, and then between the red veins will be 6 inches. So if they started out and their fingertips were touching here, and then they jumped and they got up to this red vein up here, then they would be that vertical jump height would be 12 inches. So that's how we're going to assess their vertical jump height. So our results for this trial, we have them in centimeters, because that's what we always generally want to work with. And then we can use this formula here to more accurately assess their power output, which will give us a value in watts. And then we can compare to some normative data here for different populations, and you might be able to say, okay, this athlete is similar to one of the trials where they actually end up being similar to one of the populations. So our next test is going to be the Margaria-Kalamen test. So I'll just explain it and then we'll watch video of it occurring. So we're going to be measuring the height of a set of stairs between the 3rd and the 9th step. So they'll start at the bottom, measuring though from the 3rd step to the 9th step. So generally we want to place a switch mat on the 3rd step and the 9th step. We can also use a stopwatch, is what we use in this lab. It's an even more cost-effective way of performing this, but definitely not as accurate, because as you'll see in the video, it's pretty quick, and in order to get accurate times, a switch mat is going to be ideally the way that you'll go. So you'll make sure the athlete has a couple warm-up trials, then the athlete will sprint from a standing start 6 meters away, and then take the steps 3 at a time, and the time from the 3rd to the 9th step will be recorded. And we'll do this a total of 3 times and take their best time. So now we'll watch that occurring. So we can see them stepping on the 3rd, 6th, and then the 9th step in the staircase. This staircase isn't necessarily ideal because it's a little bit crooked, as you can kind of see in our top view video. The hallway isn't necessarily even leading up, so it's not necessarily ideal, but it's what we had to work with. Okay, so our results, we have our 3 trials, and then from there we also have our vertical stair height, and then from there we can calculate our power for this, then using this formula we have. And then we can compare to some normative data that is in your lab. All right. So our last test will be the standing long jump. So the athlete will place their feet at the edge of the start line. Then they'll perform a counter movement similar to that vertical jump. And then they'll jump as far forward as possible. And they must land on their feet, and the point is measured at the back of the rearmost heel. Ideally, we would have somebody else placing a piece of tape or some sort of marker down to actually measure, just for sort of, when we were recording this for COVID safety, just to try and keep everyone as distant as possible. The athletes had a piece of tape and placed it down themselves and then recorded themselves. And so that is the standing long jump, and then the best of 3 trials will be recorded. Next, we have the results of that trial, and then we can compare to the normative data using their best trial. So key takeaways from today's lab are that field tests can be used to measure an individual's anaerobic ability. There's different tests which can be used to measure an individual's lactic, so that longer system, or alactic, anaerobic metabolism, and their capacity and their power of each system. And then these tests can be used as a part of a needs analysis to determine where an athlete needs to improve, whether their anaerobic system is a strength or whether improvement is needed. All right, and thank you for participation in today's lab. And please let us know if you have any questions when writing up your lab report. We're definitely happy to help.